greetings from Houston, Texas, quite an unusual place to be talking about cannabis, where cannabis prohibition indeed started in 1913 in El Paso. And Texas is currently on the road of the last throes of the prohibition, where the medical cannabis program has been instituted to the point where the licenses have been submitted, and the Texas government, in Texas, it's the Department of Public Safety that is overseeing this program, so state troopers, and they will issue at least 3 licenses this fall. And the topic of my talk is the Entourage effect, but I would like to, first of all, thank the organizers for having me here, the Terpenes and Testing magazine, and also Celeste from the Hemp and Cannabis Marketing Labs. And also would like to thank very much the previous and the upcoming speakers. It was very informative, and I have learned a lot, and it's very good to share these different types of information within this community. So I'm going to tell you a little bit about Cantelligence, which stands for Cannabinoid Intelligence, and it's a company that we created at the University of Houston. We're at the Innovation Center of the University of Houston, and we provide continuous medical education as well as customized curriculum training about cannabis, cannabinoids, and products to healthcare practitioners, as well as people in the industry ranging from bud tenders or cannabis technicians to investors. So we do quite a bit of consulting as well as produce intellectual property, where in my laboratory here at the University of Houston, which is not a part of the company, but the research is actually being integrated within this company and the IP, we are testing different combinations of isolated cannabinoids and terpenes, and we're testing their ability to control abnormal brain activity, synaptic transmission, but in particular abnormal brain activity during epilepsy or other neurological disorders. So by training, I'm a neuroscientist, and this is what we do here at the university in the lab, and also in the company. We're very much interested in kind of spreading the balanced, informed point of view about cannabis, cannabinoids, and cannabis industry. A lot of people, especially in the medical community, view the cannabis or the state of medical cannabis industry right now as the Wild West, and this is a view from the healthcare practitioner standpoint. The slide was created by one of our Cantelligence partners, who is a professor of medicine, Dr. Jaime Claudio Villamil in Puerto Rico. And he's actually very much interested in individualized or personalized Entourage cannabinoid treatment. The reason for it is most of the healthcare practitioners, as is illustrated in the slide, are not familiar with what kind of dosage to use, what kind of strain to use, what kind of delivery method, if it's going to be oral ingestion or a suppository. They are not certain of the efficacy of certain compounds that are found in products. They're not familiar with the formulations of the products, and they're not certain if the formulations have been standardized. And there is a really big need for observational studies in order for us to try to understand some of this Wild West and start understanding the appropriate dosages, strains, the delivery methods, and educating healthcare practitioners about this. And this is the reason why it's important, is because we have the standard way of practice that most of the medical doctors and healthcare practitioners have been trained under, which is looking into the booklets provided by pharmaceutical companies into the specific concentrations that should be assigned to their patients and adjusted accordingly to the effect of these medications, or accordingly to the unwanted side effects of these medications in patients. At the moment, there is no standardized way of treatment. There is no standardized training for medical cannabis, but we are in the process of all of this, and I'm sure that these things will be resolved in the next few years to a decade with a very good understanding of what the products are and how to use them in the proper dosages. And so what we have in this slide is a lot of what we know about cannabis is belief. It's what you believe about cannabis, and it's very difficult to shift individuals' beliefs, because a lot of times they're ingrained there for a long time due to either societal or family reasons or other such things. So for example, if we look at the believed negative effects of cannabis is that it causes lung cancer. The knowledge that we have is, well, if it causes lung cancer, there's other ways that we can ingest it. We can vaporize it instead of smoking, so we can prevent a lot of particles that are in the plant matter that could potentially be carcinogenic. But the truth is that it is a lung irritant if you smoke cannabis, but it does not cause lung cancer. And in fact, it is bronchodilator. And it may, according to some recent reports, actually reduce the occurrence of lung cancer. So it's very important that in this overlapping circles we aggregate the knowledge, and this is what Cantelligence, Dr. Jaime Claudio does, and a lot of panelists also trying to aggregate the knowledge in science, aggregate the knowledge in business extraction processes and so on, so that we could deliver the truths to the society that are less biased by beliefs and more based on the actual facts and knowledge. So when we talk about this industry, and previous speakers have mentioned, it's very important to start understanding different parts of this industry, and it starts out with the human and human endocannabinoid system. And the system is interacting with other systems, and so you have this human or personal entourage of your endocannabinoid system, the overall state of that system, whether it's naive or whether it has been stimulated with multiple cannabinoids or whether it has been stimulated with an isolated THC. There is also exogenous cannabis entourage, and this is a variety of molecules, terpenes, esters, flavonoids, cannabinoids that you will find in cannabis. And it's important that we start understanding understanding well different components of this plant and how these different components can interact with each other in producing this exogenous cannabis entourage. And that exogenous cannabis entourage will be preserved if you are, if your end product is cannabis flower. However, one of the main points of this gathering is to understand different aspects of terpenes and extraction mechanisms. And the fact is that the composition or chemical composition that you see in the plant or in the flower now can become something very different once it goes through the extraction process, through the formulation process, and finds itself on this shelf in a dispensary or pharmacy, where now cannabis products have their own entourage independent of potentially the flower, where the flower could be the same as a starting material, but the oil or the patch or the vaporizer that is produced from that now carries a different entourage of molecules. And understanding all of this in the end will lead back to the individual and back to the most effective individualized cannabis or entourage, cannabis entourage treatments. As you all know, National Academy of Sciences came out with a very powerful report which reiterates medicinal properties of cannabis in certain applications, especially being effective in treatment of chronic pain and nausea and some of the terminal disease symptoms. And one of the big calls by the National Academy of Sciences is to promote further research in cannabis, promote observational studies in medical cannabis as well as pharmaceutical cannabis communities, and to inform and to provide that information to healthcare practitioners so better medications, better standards and products can be established. Also, the same report acknowledges that there was a systematic suppression of cannabis research, especially cannabis research that has hypothesized potentially positive therapeutic effects of cannabis. So this is a publication that you can find online, hopefully download as well. The science tells us that we have endocannabinoid system. Endocannabinoid system is a major body regulatory system. It regulates homeostasis of the body, and it consists of the cannabinoid receptors, CB1 and CB2, as well as endocannabinoids. The two most prevalent human endocannabinoids are anandamide, or AEA, and 2-AG, or 2-arachidonyl glycerol. And together with their synthesizing enzymes and their hydrolyzing enzymes, and with these endocannabinoid molecules and receptors, they comprise a major system in the body where CB1 receptors are predominant in the brain, in the heart, and in the digestive system, and CB2 receptors are mostly expressed in the organs that are responsible for amassing immune response, such as spleen, and are expressed in many different immune organs and cells. It's a system that gets activated very early on. If you may think that CB1 receptor activation by THC induces munchies, and THC is not endocannabinoid, it's a phytocannabinoid that comes from the plant, but anandamide and 2-AG also bind CB1 receptors. So it's very likely that early postnatally the hunger comes not from the reward system, which is dopamine, because reward system kicks in after somebody has eaten, but hunger may be promoted, in fact, by endocannabinoids and endocannabinoid system, in part through the activation of CB1 receptors. And so it's important in the newborn suckling process. It's also a system that is modulated, can be modulated to control pain and stress, and it itself modulates stress. It's involved in immune function and appetite control, in memory, where some aspects of cannabis plant like THC could impair short-term memory, and other cannabinoids like cannabidiol or CBD could improve the memory. It also affects inflammatory processes by reducing inflammation, and is involved in thermoregulation as well as other effects. And what the bottom of the right shows here is that anandamide, which is endogenous cannabinoid or cannabinoid produced by human bodies, is very similar in structure to tetrahydrocannabinol or THC, which is exogenous cannabinoid produced by the plants. The way the cannabinoids work on the receptors is they bind to these receptors, often represented, this binding with a key and a lock kind of fashion, and we are trying to understand how these keys turn the lock. And there are many locks on the doors, and these doors are the receptors, and so there's many keys and many different openings that could cause the opening of this door or activation of that door to a lesser or greater extent, as would be the case with CB cannabinoid receptors. In the brain, and the discovery of endocannabinoid system really came from the neuroscience studies, where motivation was, of course, that it is an intoxicating substance that causes euphoria, therefore the effect really probably primarily, if not solely, lies in the brain. That was some of the early hypotheses. What is depicted in the slide on top is one of the neurons. It's called presynaptic neuron. At the bottom, it's another neuron. It's called postsynaptic neuron. This is an example of how brain cells communicate with each other. And they communicate with each other through these synapses. And the top cell has these yellow vesicles. These yellow vesicles are filled with neurotransmitters. Neurotransmitters such as glutamate is most prevalent. It's the main excitatory neurotransmitter in the brain. And when there is excitation of the cell on the top, then you can see these yellow bubbles, which are little glutamate molecules, then get released. The yellow circles fuse with the cell's plasma membrane, or cell's wall, if you may, and they release these neurotransmitters that are glutamate. Glutamate then activates glutamate receptors postsynaptically, and there is a response. But in certain instances, there's too much of this glutamate produced, and we need to control the production of glutamate, because too much of glutamate can lead to what we call excitotoxicity. With too much glutamate released between the cells will end up in killing cells or turning on programmed cell death inside these cells, where they will lead to their own self-destruction, so to speak. And so when there is increased signaling of this glutamate, then you have activation and you have on-demand production of endocannabinoids. Endocannabinoids get synthesized postsynaptically. Notice that they're not stored in these round vesicles like your other neurotransmitter molecules are, but instead they freely cross through the plasma membranes, and in what is called a retrograde fashion, or in this negative feedback-like fashion, then the endocannabinoids can activate CB receptor. CB1 receptor in particular, we focus here is on the neuron, but there's also other types of cells that are called glia that are very important in nerve and brain function, and those cells as well have cannabinoid receptors. When an anandamide or 2-AG activates CB1 receptor, the CB1 receptor is linked to what we call G protein. So it's a G protein coupled receptor, and it's very widely expressed. Perhaps the most predominant G protein coupled receptor in the brain is CB1 receptor. Activation of that receptor will then lead to shut down of calcium influx on this first neuron on top, and this will prevent now or balance out how much glutamate, how much of those yellow molecules are now being released between the two cells. And so in this way, the cannabinoid system balances the level of excitation in the brain, but it also balances the inhibitory neurotransmitters, the level of inhibition. And in addition, it also interacts with many other neurotransmitter systems in the brain. Anandamide and 2-AG are both partial agonists. 2-AG will bind CB1 and CB2 receptors with the same strength or same affinity, but it has a stronger binding to CB1 and anandamide. And when anandamide binds these receptors, it prefers to bind to CB1 receptor. The functions that are associated with the activation of these receptors is control of analgesia, inflammation, oxidative processes, cell tumorigenesis or cell proliferation, as well as neural function, like I just talked about, the balancing of the activity in our neurotransmitters. They're partial agonists, so they just partially activate that door. And when we talk about full agonists are sometimes enough. One key and one lock is enough to activate the door to a great extent. And so our endocannabinoid systems balance excitation and inhibition, and we ourselves balance excitation and inhibition, and we basically react to life. We adjust ourselves, we adjust our brains, and we adjust our behavior. Now, as I mentioned, endocannabinoid system, it's not only regulated glutamate and GABA or excitation and inhibition, it's actually regulating other neurotransmitter systems that are important in the brain and body regulation. This is an expression pattern of CB1 receptors, which illustrates, again, how vastly these receptors are expressed in different parts of the body, and how certain parts of the brain, for example, like prefrontal cortex or cerebral cortex or basal ganglia or hippocampus, very important parts of the brain that control motor activity, that control emotional response, learning and memory, are a part of this endocannabinoid system. And I always point out that endocannabinoids and endocannabinoid system, CB1 receptors, are not prevalent in the parts of the brainstem here that are responsible for vital functions such as breathing and heart rate. And that is the difference between endocannabinoid system functioning and cannabinoid functioning in effect, the physiological effect of cannabinoids on the body as compared to opiates. So opiate receptors will be very highly and densely expressed in the brainstem areas that actually are controlling vital body centers such as breathing and heart rate. So endocannabinoid system gets engaged when you have stress, when you have trauma, when you have too much activity, we call it hyperexcitability in the brain, and when there is inflammation. In response, endocannabinoid system tells the body to eat, to rest and sleep, to forget, and to protect itself. And forgetting is an important part of the survival, and as well as protecting the neural mechanisms and the balance of these neurotransmitters. Controlling this neurotransmitter balance is beneficial to the brain and can be neuroprotective, protecting the cells from overexcitation and from eventual death in overexcitation. Endocannabinoid system, or is it multiple systems? The fact of the matter is that endocannabinoids, when we talk about endocannabinoid system, we talk about CB1 and CB2 receptors, but endocannabinoids in the system interacts with other receptor neurotransmitter and receptor systems in the brain and the body, such as serotonin. Serotonin is controlling the mood and appetite intake and some sexual behavior. TRPV or tripvanilloid receptors that are pain receptors, they're also activated by endocannabinoids. Peroxisome proliferated activated receptor, PPAR, which is involved in cancer growth or proliferation of cancerous cells. It can also affect ion channels on those plasma membranes that we briefly mentioned. It's a system and receptors and endocannabinoid molecules that are expressed in distinct body organs, and impairments in endocannabinoid system or regulation of endocannabinoid system with cannabinoids can have a very distinct effect on very distinct syndromes and disorders that range from epilepsy to inflammatory bowel syndrome to inflammation and such. Perhaps inflammation could be the underlying sort of a cause or a common factor between many of the disorders in where cannabinoids show beneficial effects. I won't go into details of that, but we know that most of the cannabinoids and terpenes are located in these beautiful trichomes. Over 95% of cannabinoids expressed by the plant, as was mentioned by one of the speakers earlier, is acidic cannabinoids. They're non-psychoactive and they turn into neutral forms from THCA, for example, into THC with heat or decarboxylation or some other manipulations. There are many different cannabinoids, over 115 different cannabinoids, major and minor. Major are the ones that are expressed to a greater degree, such as THC, CBD, CBG, and then there are minor cannabinoids that are expressed to much smaller levels or percentages. And perhaps this is just the beginning of the cannabis evolution and cannabinoid evolution, because as we see some of the really spectacular landrace strains that are locked up in remote areas of the world can sometimes have expressions of these minor cannabinoids that are much higher in percentage actually than these major cannabinoids. Terpenes are a very important component. We have many different terpenes. Limonene, caryophyllene, and linalool are among some of the few, to mention some of the favorite ones that I think about. And it's a diverse set of phytocompounds that you find in cannabis sativa. Terpenes, phytocannabinoids, flavonoids, amino acids. THC has many different effects. The main psychoactive agent of cannabis is anti-inflammatory, it's neuroprotective, it's analgesic, appetite stimulant, it's antiemetic. But we also have to remember that if you are not working, or the THC is not working well with your body, or if there's a big amount of THC that has been taken, it can cause negative effects such as increased heart rate and panic attack, which are temporary and are not deadly in any case. Cannabidiol, or CBD, is probably the champion or therapeutic champion of cannabinoids, and is very highly studied currently. And so there's many different effects that cannabidiol does, but it has antipsychotic, neuroprotective, anti-cancer properties. And interestingly, CBD a lot of times can counteract negative effects of THC. So the way that CBD works, it actually prevents the breakdown of endocannabinoids, increases endocannabinoid availability. It allows for rapid crossing over blood-brain barrier. It's non-psychoactive, non-intoxicating, counteracts THC, such as paranoia, anxiety, and panic attacks that could be caused by THC. There is sufficient evidence in human epilepsy on CBD as well, in diverse case studies. There's an overview of cannabis and epilepsy, and I could just give a whole separate lecture on this. But what is very interesting, and I'd like to point out, is when cannabidiol is being used in these studies, either open label or stage 3 clinical trials. It's an isolated cannabidiol that is being used, and it's showing a reduction, median reduction of 36% in motor seizures in patients. But some of the recent data is actually really interesting with extracts that show that there is up to 75% reduction in seizures, and there is basically a greater number, if I'm not 30 found ineffective, 1750 experienced reduction in seizures, and 28% experienced essentially 90 up to 99% reduction in seizures, and 10% experienced a complete clinical response. Of course, this is a survey study, so it's difficult to tell, but what is happening there is that in the pharmaceutical world, I'm very briefly just going to talk about this, you have cannabidiol, which is an isolate, and in the medical cannabis world, you have extracts, and you have multiple extracts that contain different cannabinoids and different terpenes. And again, this is another study in Israel reporting that there was 18% up to 100% reduction, and another 34% reported up to 75% reduction in seizures. So there's going to be some very interesting science and studies coming out once these natural extracts and combination of cannabinoids and terpenes are being used. As we know, terpenes have their own biological properties. Limonene, D-limonene is a potent anti-cancer anxiolytic. Myrcene, which you will find in hop and mango, is a potent anti-inflammatory, analgesic, and anxiolytic. Alpha-pinene is an acetylcholinesterase inhibitor, which acts very similarly to essentially Alzheimer's medications. Linalool is an analgesic, and beta-caryophyllene is anti-inflammatory. And so as we talk about different parts of this plant, it's very important for us, again, to remember that, I'm trying to bring up the slide, to remember that this is, again, all about supplying and recommending the best product to patients. So we have to understand the entourage in the body, the entourage in the products. We have to educate doctors and nurses, healthcare practitioners. They have to communicate to their patients about the observation, about how different effects of cannabis or the entourage components are working for them, with them, or against them to improve basically the individualized treatment with cannabinoids. These are some of our partners. I think I'm running out of time, and if you'd like to contact Cantelligence or myself, here is the contact information. Thank you very much. I'm going to see if there's any questions. Let's see how I can visualize these questions. I know I'm almost at my maxed out time. Are there any questions? I think
we do have one question for you.
Okay, Riddick said, as someone new to the field, where would you suggest I can read up more? Well, that's a great question, or it's Riddick Gosh that said that. You will have to find books on Amazon, and there are some very good books on Amazon. Cannabis Pharmacy is a very good book that I would recommend. And multiple other books that you will find on Amazon. Unfortunately, textbooks do not teach about endocannabinoid system or the cannabis components and the entourage effect, and so you will have to seek out a lot of that information. Make sure it's from the reliable sources, and make sure that the books that you read are by accredited people and have been reviewed by other accredited people and commented on by other folks as well. You could try our website, cantelligence.com. This is a shameless plug, but you will find some useful information there, and we do put out really interesting articles that are relevant to cannabis science as well as cannabis industry. And of course, PubMed, if you have access to what is called public access articles. So you can search for public access articles on Google, and a lot of times you may gain access to some of the literature that is available online. So I hope that answers this. Now, it says NIH has over 2,000 articles and peer-reviewed papers on cannabis cannabinoids and the cannabinoid system. Yes, absolutely. So not everybody has access to these articles, but if you go through the public access system, or some of these articles are published in public access magazines, then you should be able to access them as well. Yes, and so if there is no further questions, I thank everyone.